Warning.
Today we're going to be talking about black widow spiders. These spiders are super cool and they have a distinctive red hourglass on their abdomen. But what really makes these spiders special is that they have one of the most painful spider bites in the world, possessing a potent neurotoxic venom, α-LTX, with a strangely beautiful final tetramer structure. I'm going to be breaking down this study by these people and showing how structure and function in macromolecules and proteins are directly linked, and what it takes for a scientist to actually figure out how a molecule like this works. I've really enjoyed reading this study, as these scientists were really able to capture some beautiful results, and I believe the manner in which this neurotoxin works is truly just amazing. And so I'm really excited just to share this with you, and let's just get right into it. The potent neurotoxic venom of the spider actually contains 7 different latrotoxins, most of them targeted at insects, but some targeting crustaceans and one targeting vertebrates. This study primarily focuses on the α-Latrotoxin, which is the one that affects vertebrates, like humans. Alpha-Latrotoxin contains 4 monomers and therefore is a tetramer. Once it becomes a tetramer, it becomes easy for this molecule to form and attack the cell, releasing massive amounts of neurotransmitters like calcium 2 plus ions and sodium plus ions. This is an example of the difference between monomers, dimers, trimers, and tetramers. A monomer is just one part, a dimer is a connection of 2 parts, a trimer is 3, and then a tetramer is 4. So if we can see that our full molecule is actually a tetramer of G-shaped monomers, and once that comes together, that's when we were able to have this coiled coil stalk actually unravel and permeate the cell membrane. And they've also provided us with an actual model of how this works now, because we have enough data to actually understand how it works. And now I'll try to explain it. So this process starts in the cell of the spider where these proteins are actually produced. The first thing that has to be done is the cleavage of the furin-like protease. And this will allow these alpha-LTX molecules to actually tetramerize a little bit better, and they can either tetramerize with the aid of receptors or without the aid, but they tend to tetramerize better when in the presence of a receptor. But once they are bound to a receptor, and the introduction of a calcium 2 plus ion makes the coiled coil stalk uncoil and makes this disulfide bridge a permeable area for these positively charged ions to move through the lipid membrane. And I'm going to show you now the supplementary figure. So now we're going to see the transformation so this is when a Ca2 plus ion is introduced, or it's trying to attach itself to something, right? So we see this unfurling of these coiled coil structure, and we see all these helices, and it creates this negatively charged path for all these positive ions to move through into the cell. And of course this will wreak havoc on your neurotransmitters, and everything in your body will go haywire if you get enough poison in your body, which is highly unlikely. Now, all of this research was made possible by the invent of the cryogenic electron microscope. These electron microscopes are super powerful, and they have more ability than any other tool in history to get this up-close look at these molecules. And here's something else that's cool. This is a flowchart from that top left box to those 2 pictures in the bottom. Scientists use a combination of AI technology and huge electron microscopes in order to compute grand images like this, and it's truly amazing to see how they even do something like this. Here's some additional beautiful figures that I just wanted to include with this project, because I can't explain everything. This is complicated science, but it is really cool, and it is really beautiful. And here we have estimates of the charge and how hydrophobic these places are, It's very cool to see. The researchers also found out that there's another chemical that's pretty similar. But overall, this project was very fun to do, and I learned a lot about how proteins work, how they're made, and how they're applicated, and I hope you learned a little bit too. Have a good one.